The first company we have on board is Neuropeutics, who is pioneering a mechanism that could slow or halt diseases where current drugs only manage symptoms. Mark and his team are developing therapies that reverse protein aggregation, the root cause of many neurodegenerative diseases. Mark, over to you.
My name is Mark, and I'll be walking through what we are building here at Neuropeutics. The unfortunate reality that we live is that every minute around the world, 2 people are being diagnosed with a neurodegenerative disease, and they're going to doctors to be told that there is very little that can be done for them. And so we co-founded Neuropeutics, a pharmaceutical company developing targeted therapies to extend the neurodegenerative disease patient survival, to improve patients' quality of life and extend their survival. We are first targeting amyotrophic lateral sclerosis, or ALS for short, and it's really a fatal neurodegenerative disease with no effective treatment. Each day, 14 people receive their diagnosis, and they're being told they only have 2 to 5 years to live. And over these years, they start losing their ability to walk, to talk, to eat, and then finally, their ability to breathe. And the current treatment options are highly ineffective, only extending patients' survival by an average of 6 months. What happens normally is that neurons, the cells of the brain and spinal cord, are what control our muscles and our movement. But what happens in disease is that these neurons are dying, which leads to muscle atrophy and progressive paralysis throughout the body. And one of the main drivers for this phenomena here is this protein in green that I call TDP-43. This protein is found in all of us, and it's a very crucial protein that carries very important functions. But in disease, this protein leaves its normal location and forms these toxic clumps within the cell that leads to the neuronal death. And really, there's 2 disease mechanisms at play here. The protein is no longer in its normal location, and so it can't do its normal function. And also, these toxic clumps within themselves are toxic. For decades, people have been trying just to clear these protein clumps, and there is no benefit to patients. And so at Neuropeutics, we have developed a first-in-class small molecule that doesn't only just clear these aggregates, but we're actually reversing these protein clumps, allowing the protein to go back inside where it belongs to carry out its normal function again. Our molecule is highly efficacious, that within only 2 weeks of treatment in an animal model, we're able to reduce disease pathology by 30%. And this is shown here in an image from a mouse brain. All these red dots are the toxic clumps that's reduced upon treatment. The reduction of this TDP-43 clumps is very well established to lead to a restoration of motor deficit, prevention of neural loss, and more than doubling of the lifespan of the animals. We are really targeting the pathology at its root. When we look at the other 2 FDA-approved therapeutics, they target secondary mechanisms, things that are arising due to the initial presence of this TDP-43 protein clumping. And so it is no surprise that when we look at their efficacy is that Edaravone actually has no effect at all and is expected to come off the market in a few months, with riluzole extending patients' average by 6 months, with a few rare case studies showing up to 19 months. But us, by targeting pathology at its root, we're looking to add years to patients' lives. We are first addressing the ALS patients in the United States, which is a market valued at $1 billion, where a single ALS therapeutic per patient per year can cost upwards of $158,000. Globally, that's a $3.8 billion market, and globally, and as we start expanding into other neurodegenerative disease indication, we're looking at a $40 billion market. JRMS-22 is our most advanced asset. We have a novel mechanism of action. We are patent pending at the PCT stage with national examination starting in the next 2 months. We have the IP license from the University of Toronto. We have promising preclinical efficacy. And what's really interesting is that with our molecule, we have our target, we have unlocked a mechanism that's able to show therapeutic benefit in models of ALS, but also against alpha-synuclein in models of Parkinson's and tau in models of Alzheimer's. We also have favorable preliminary metabolism and toxicology studies. We also have identified 2 novel targets that we're looking to build chemical programs for them as the company grows. Our team is really the team to advance this to the clinic. Collectively, we have over 150 years of expertise in ALS research, diagnosis, clinical trials, and business development. I've spent roughly the past 10 years really developing the assays and advancing this small molecule alongside my co-founder, Dr. Janice Robertson, who's the Canada Chair of Research in ALS and the highest publishing author in basic science ALS in the country. To date, we have raised over $1 million in non-dilutive funding from the company spin out about 2 years ago. We have won multiple national and international pitch competitions, but our biggest win yet was in January of this year, where we announced a significant partnership with LifeArc. It's a multi-million-dollar partnership through which we are receiving in-kind CRO services, and we have a dedicated team of 10 scientists who are working on advancing the chemistry. So what I mean by that is that we have the molecule, but we really need to take this molecule and turn it into a drug format that's able to ruin patients, and that's what LifeArt is leading the chemistry, and we're leading the biology. In May of this year, we've also received the AbbVie Biotech Innovator Award, and through AbbVie, or with AbbVie's help, we are meeting with their R&D, neuroscience team, and venture teams to really ensure a successful IND application. So right now we're optimizing our molecule. We are looking to have our IND filing by 2028 and initiate clinical trials by 2029. Currently, we're raising a $1.65 million pre-seed round led by TAP. We are using this money to achieve these inflection points. So we've already achieved blood-brain permeability, which means the drug can now reach the brain. We have reached novel matter of composition, which allows us to expand our patent and create a portfolio of patents. We are looking to now really have this into a lead candidate selection, show efficacy of this new molecule in humanized mouse models, and then safety in larger animals before IND filing. Every dollar that we are raising will be matched at least 1-to-1 through other non-dilutive programs. We only have $245,000 left in our round. We are raising this on a convertible note with an $8 million cap, 25% discount, and 7% interest. This is our 8-year roadmap with envisioned exits at end of 2029 upon IND approval or upon completion of a phase 1b/2a in 2030. And so I hope that I've convinced you that we are addressing a huge unmet medical need. We have a first-in-class therapeutic that's scientifically validated with strong IP. We have a clear competitive advantage by addressing the disease at its root. And we have a strong team with a track record of success in pharma. So join us on our journey so that one day when a patient sits down at a doctor's clinic, they're being told that they have ALS, but they're also being told that there is now a new effective treatment for them, a treatment developed by Neuropeutics. Thank you very much for your attention.
Fantastic, Mark. Thank you very much. Just under time, which gives us more time for question and answer. I'll leave maybe 20 seconds for people to kind of whip up a question and put it in the Q&A section. If not, we do have a couple of questions that we can kick off with. All right, so maybe as people are thinking of Q&A, Mark, a question for you. How are current ALS approaches failing patients today, and where does your mechanism meaningfully change that outcome?
Yeah, so most of the ALS therapeutics are either addressing symptomatic treatments or they are, addressing secondary disease mechanisms. And neurodegenerative diseases are really complex, and I'm by no means saying that a single therapeutic will solve it all. But we're going upstream, we're targeting the root of the disease that can really prevent all of these secondary mechanisms from happening.
Fantastic. And maybe take everyone through your future plans. How soon do you plan on an exit? How does an exit look like for you within this space?
Yeah, so historically, exits have usually been after a phase 1 or a phase 2 trial. But especially now in ALS, we're seeing a lot of shift or a bit shift where a lot of assets are being either acquired or out licensed to big pharma at the IND level. And I think one of the most recent examples was a deal by Lilly for an asset that's for a very specific form of ALS that's familiar about 1% of the ALS population. And such exit was, over roughly $500 million USD. And so the exit most likely will be an out licensing or an acquisition by a big pharma.
Fantastic. And we have a couple of questions in the chat, Mark, so I'll ask a couple of those. Could you say more about the 1-to-12 match?
Yeah, so the 1-to-12 match is through our LifeArt agreement here. So LifeArt is really putting up most of the funding for this program. It's an extremely friendly deal, and that's thanks to our advisors who have helped us negotiate this deal. And they're really putting up all the cost for the chemistry, for the safety studies. And in Toronto here, we're only paying or using the pre-seed round just to test the biological validation of these molecules that will bring us to IND.
Fantastic. Mark, how many competitors do you have in Canada? And yes, this presentation will be shared after in the recording.
So in Canada here, there is only one group out of BC that is seems to be addressing ALS. They do have a completely different therapeutic approach. They're looking at using an antibody, and antibodies inherently are a lot more expensive, are a lot more difficult to manufacture, a lot more difficult to even reach the brain. And so they're also at the discovery stage, so they're a little bit earlier than we are. But it's an approach that again only clears out the clumps. It doesn't address that the protein needs to resume its function again. And so it's a very different approach, and it's at an earlier stage than we are.